TBRS was first identified. There was really just one research group that was interested, and that was through the Childhood Overgrowth Study in the UK. And since then, the Tatton Brown Rahman Syndrome community has built a collaborative research network. So in the beginning, it was Dr. Tatton Brown's group and some folks at WashU and at Baylor College of Medicine. And then during the pandemic, we got a seed grant from NORD to fund building our collaborative research network. So we started basically just contacting anybody who had published on DNMT3A, or if we were lucky on Tatton Brown Rahman Syndrome. That wasn't many. There is a lot of interest in our gene, DNMT3A, because although our children are born with a germline mutation, usually de novo, it's usually not inherited. People acquire mutations on DNMT3A in the aging process, the somatic changes. And Dr. Lay at Washington University had previously discovered that those somatic changes to DNMT3A were a main driver in AML, the development of AML leukemia. And there's also links to somatic changes in DNMT3A and cardiac disease. And there's a lot of interest. It's a methylation gene, so there's a lot of interest in epigenetics right now. So there were actually a lot of publications on DNMT3A. So we reached out to everybody who had published and invited them to meet with us, and we told them about our rare disease if they hadn't heard about it before, just so that we could see if the progress they make in their work could affect treatment development for our families. So we've built our collaborative research network to over 230 clinicians and researchers. And we've also developed a lot of research tools to help engage researchers in accelerating the identifying therapeutic options for TBRS. So we have a patient registry, which is a natural history study. We have a biorepository where we collect patient samples, and we link the data from the registry with our patient samples by use of something called a CRID. It's an identifier. So it's, the data that we can release is all de-identified, but you're able to tell, like, oh, this patient sample is linked with this information from the patient registry. We've funded the development of iPSC lines. We have real intimate knowledge of the scientific community and of our patient community. So when people approach us and say, oh, is there a scientist with a mouse model? We can tell them about, if the scientists are comfortable with us sharing, where those mouse models are and if that scientist is willing to share it. So we're doing our best to, have all the tools needed to help really accelerate research, and we have some wonderful scientists working with us to do that.